Hello. Today we're going to talk about the process of starting with the plasmid DNA and ending with a pure recombinant protein. The whole idea is to look at and learn how to become familiar with the process, starting with this plasmid DNA, expressing, purifying, and then characterizing and processing the protein for other work. We're going to focus on his tag affinity purification. There are other affinity tags to look at, but right now this is what we're going to focus on. So let's start to talk about the overall workflow. We're going to assume that you have a plasmid, a malate dehydrogenase or another protein. It's going to be affinity tagged with a 6 hex motherfucker. All right, so let's talk. So the experimental workflow. The idea is you've already obtained or made yourself some gene, inserted it into an expression plasmid. So now you have yourself a plasmid with some gene ready to go to express and make protein in bacteria. There's all sorts of issues you need to be concerned about when thinking of this. This is for a kind of a molecular biology course or techniques, thinking about the transfer RNA, the codons, the rare codons. Maybe you took a plant or a human gene and stuck that into a bacteria plasmid vector, but the transfer RNA or the tRNA isn't at high enough level to get a good expression. So there's codon optimization and things like this. But we're going to assume you've got everything cloned into the right kind of plasmid ready to go for expression into a bacteria or E. coli cell. We're going to look at the expression of this. What does it take to get good expression? Talk about inclusion bodies a little bit. Talk about the size of culture, how to change culturing conditions so you can get the most possible soluble not inclusion body or cytosolic expression level in these bacteria. Then we're going to talk about how to break the cells, centrifuge the cells, and then purify the proteins. And then finally, once the proteins are purified, we need to characterize them, look at how much protein's there, dialyze them, maybe cut off the affinity his tag, things like this. So this is the overall workflow. We're going to do this video in 3 steps. So let's get started. Expression. So the whole idea is you want to grow up a culture, put your plasmid in the bacteria cells, and have those bacteria cells act like little factories to make lots and lots of your protein. All right. So what do we need to get started? Well, it depends on your materials. For folks that are working with MDH in the MCC world or other courses that are using MDH, these have been cloned into either one of 2 kinds of bacteria plasmid backbones, a PQE60 for the watermelon MDH and what's called a PET vector with most of the other MDHs and other proteins like citrate synthase and PepsiK and malic enzyme. And in this case, we have a his tag on the C terminus of all the MDHs because putting something on the N terminus of malate dehydrogenase causes a problem with how that protein regulates and functions. And some of the other proteins, the his tag will be on the C terminus. So you've got your DNA. Now we need to get the DNA inside the bacteria through a process called transformation. But what bacteria? How do we do this? Do you start with that DNA, the freshly transformed cells? You already have them spread out on a plate so you can pick one of those colonies and go. That's what we're going to talk about. So in general, because we're talking a lot here about malate dehydrogenase with either the watermelon or some of the other sources, we have lots and lots of different isoforms. MDH is cloned from different species, from human, plant, watermelon, all sorts of pathogens, you name it. They typically fall into 3 different categories: really strong expressors. Human malate dehydrogenase 1 or cytosolic MDH is an example of a very strong expressor. The watermelon MDH is also a very strong expressor. We have mid-level expressors, the mitochondrial MDH from humans and others are about there. And then the mid-level expressors, citrate synthase and some of the other MDHs. So based on whether we're looking at low, high, or mid, or really strong, depending on the culture conditions you do, you can expect a rough estimate of concentrated protein. If you do everything right and you purify the protein and cut your samples in a way to get high yield, you can expect to get anywhere from a 0.2 or so mg per mil solution all the way up to a 5 mg per mil solution for the very strong. Again, that would be like the human MDH1 or the watermelon MDH. Now, how much total protein you get, of course, depends on how big your culture. And this table is, it was prepared using a number of experiments where we tested on a number of different MDHs. So it can give the user a kind of an idea, how much protein do I need? So therefore, how many mils of culture do I need to make? So if you have a really poor expressor, you might have to have a really big liter or 2 liter of culture if you need a lot of protein. If you're doing a strong or very strong culture, you might only need to do 100 mils or 250 mils or so of culture to get 10, 20, 30 milligrams of purified protein at pretty high protein concentrations. For those of you who are going to be purifying various mutants of MDH or other proteins, you need to test this each time, because for some reason, some of these mutants, which doesn't make any sense if all you do is change 1 or 2 amino acids, can really impact the expression level of these proteins. But we're going to talk about how to look at the expression level and how to adjust to take care of these low expressors. So the first thing you want to learn when you're working with plasmid DNA and sticking that or transforming that bacteria, that DNA into bacteria, is what strain of bacteria you're going to use. Because bacteria are not all the same. There's so many different strains of bacteria out there in the world, but for biochemistry, molecular biology, biotechnology, we're really looking at 2 main strains or 2 groups of bacterial strains. And the first thing you need to know is, well, what are you going to do? Are you going to want to take your plasmid DNA, put it in bacteria so you can then put a little glycerol in a culture of this stuff and throw it in the freezer for later to make more DNA from? Do you need to save that DNA? Do you need to make more DNA? So maybe you want to do some mutations or some other changes to the plasmid you received. Well, then you want to pick or select a bacterial strain that's going to be suitable for working with DNA. In other words, to make DNA stable. And DNA isn't unstable, but you don't want to put your DNA in a bacterial cell where the bacterial cell will start chopping up through endo or exonucleases your precious DNA. And so you want to use something like a DH5 alpha, a top 10, an XL blue, or an alpha select. These are different strains or modifications of various strains of bacterial strains that have a lower amount of endonucleases, so the bacteria doesn't so easily cut up that stuff. You get better yield. There's less recombinases, so that way your plasmid stays intact, doesn't get inserted into some genome or some other chunk of DNA gets inserted in your plasmid. That gives you the ability to have what we think of as the stability of the plasmid. It's not getting cut up, it's not getting moved around, it's not getting ejected from the cell. And again, DH5 alpha is one of the most common strains, top 10, XL1 blue, and alpha select. Now, if you want to take your plasmid DNA, stick it into bacteria so you can make a whole bunch of protein, then you want a different strain. And it's really important to know what strain. That means when you're doing the work on plates or glycerol stocks or other solutions, you always have to indicate not just the kind of plasmid you have, but the strain of bacteria you're working with. Because a lot of the strains of bacteria that are like DH5 alpha, they're okay for doing protein expression, but a lot of them don't do as well as other strains. And the same can go in the reverse. The strains of cells, and I'll talk about in a second, for expression of protein, they're horrible for keeping stable plasmid around. It will get chopped up by exo and endonucleases, restriction enzymes, things like this. All right, so for the strains that express protein, what we're looking for is various strains where people have knocked out or deleted genes for proteases, because the last thing you want to do is grow up your bacteria culture, have a whole bunch of protein, and by the time you lyse out the cell and start purifying your protein, excess proteases have chopped up your protein into little bits. And so most of these proteases come in outer membrane enzymes, and then the ion protease. So intracellular and membrane associated proteases. And a lot of these strains for expression of proteins have already gotten rid of those genes. They're not so good for DNA fidelity because they have a very poor restriction and methylation system, and that's how the bacteria protects from the nucleases that happen. So a lot of DNA that gets put in these cells will get chopped up and cut up. And so, you know, it's not a good way to maintain your plasmid for a long time. So another thing to think about when you're culturing, especially for expression and not so much for DNA maintenance or making new DNA, are a couple of what we call helpers. So some of these plasmids are just fine all by themselves. The PQE60 is an oxygen backbone where we put the watermelon MDH into. That plasmid has a promoter that works with the bacterial promoter system and everything's good to go. You don't need to worry about anything else. But there are a number of other plasmids that have a promoter coated upstream of the start point that are for something, let's say the T7 RNA polymerase. Bacteria, especially the BL21 kind of bacteria, do not have that polymerase. So the polymerase that those bacteria have won't bind to the plasmid and start making messenger RNA. But the folks that generated some of these plasmids thought about this and said, well, we want to put a polymerase start using a polymerase like the T7 RNA polymerase that's really active. This is a fast enzyme. It's high throughput. It can actually go through the whole plasmid, to be honest with you, and make a ton of messenger RNA. And that messenger RNA is going to be used to make a whole bunch of protein. The only problem is if you put a promoter in the plasmid in front of your MDH or other protein for the T7 RNA polymerase, the bacteria doesn't make that polymerase and you don't get expression. And those are the DE3 plasmids. And the PET vectors are an example of this. So you need to have a bacteria cell that's appropriate for making protein like the BL21 cells, but also has the DE3 designator, okay? And so this is the helper we're talking about. So when you see in comma or in a cell strain marker DE3, that tells us that the lambda DE3 lysogen has already been included in these cells, and that means that these cells are now through the DE3 lysogen expressing the gene for T7 RNA polymerase. And so in this case, these cells that say BL21 DE3 means it's BL21, so low proteases, and DE3 now has the gene to make the T7 polymerase. So the T7 polymerase can bind upstream of your start point and get that really robust, strong protein expression of your plasmid that you're using. And the PET expressors for a lot of the MDH clones that we're using need this T7 helper. All right. What's interesting too is the expression on this DE3 plasmid is driven by IPTG. So later on, when we start talking about inducing protein expression, we don't make the RNA polymerase until we add IPTG. So we're actually turning on the polymerase, and that polymerase drives here. We don't have an IPTG activator on our PET vector itself. It's actually driving the RNA polymerase to do its job. Another helper, if you will, is the P-lyse system. And so in some cases, there will still be a little bit of T7 RNA polymerase being expressed, binding, and then making a bunch of different protein that's kind of a background or basal level, even before we had IPTG, because this lactose polymerase is a leaky expressor. There's a little bit of sugars already in the cells that'll bind and get this thing started. And if you're making a protein that's deleterious to the cell, it's toxic, or you don't want a lot of protein until it's time, you need to somehow to shut this system off. And that's what the P-lyse system does. Excuse me. So what happens is the P-lyse system, is an additional plasmid besides treated with the lambda DE3 lysogen to add the DE3. There's another plasmid in these cells before you add your plasmid that expresses a lyse S or a lyse E. These are T7 specific proteases. So these are proteases, and while before we said we don't want our bacterial cell line to have proteases, we're talking about general proteases that'll attack all proteins, including the MDH or other proteins you're making. But in this case, the P-lyse plasmid is a creating or expressing a protein that is very specific for the T7 polymerase. So any background or basal level expression of the T7 polymerase will get chewed up immediately by the constant expression of this protease driven by this P-lyse plasmid. So therefore, it's very silent. But once you add IPTG, there'll be so much of this present, not enough for the P-lyse to cut up and block from being used, then you can get your Pet28 or other PET expression vectors to go. Another modification to some of these expressors are if you have really low expressors for whatever reason, your messenger RNA is being degraded or you're just not making a lot of protein and you want to make more, well, then you can find a BL21 bacteria strain that has an additional gene added to this. And this is the RNE gene, or the gene that codes for ribonuclease E. And so what happens is they don't actually put the RNE gene in, they take RNE 131. So this is a mutation that has a truncated RNE and blocks the degradation, or doesn't allow the degradation of RNA. It stops the normal degradation of RNA that already happens. So that means your RNA that the Pet28 expression is making has a longer half-life. That means you get more protein. Another thing you want to think about is what strain. As I said before, you have sets of strains for DNA, production and storage, and then we have protein expression. We already hinted at the BL21. These are gram-negative strains of bacteria that initially came from the human intestine. They're the most commonly used or variants of the BL21. A lot of bacteria that are used in biochemistry, molecular biology, and other molecular life sciences are either K strain or the B strain. The K strain came from a lab in France and then kind of got handed out and derived from there. And the BL21 strain, or the B strain, came from early phage studies used and isolated from human intestine. BL21, they're pretty safe to use because they've knocked out a couple of genes that makes it very difficult to actually get into humans and propagate. But what the BL21 cells don't have is that T7 polymerase. So when you see a BL21, and if you see strains that say BL21 parentheses DE3, they're not the same thing, because the DE3 means there's an additional set of genes in there to drive the T7 promoter. So BL21 and BL21DE3, one's a variant of the other, but they're not the same thing, and they cannot be replaced easily back and forth. And then if you look at different biotech companies, there's a ton of different strains out there that support protein expression, and here's a few of them. To make our histag protein, there's at least, I don't know, 6, 7, maybe even more different variants of the BL21 strain. We'll just focus on the 4 that you see here. So there's the tuner strain. So the tuner strain has, there's a couple lactose permease mutants that these strains have. So IPTG is an analog of lactose. Lactose will be transported into the cell and then used to drive the lac operon. IPTG doesn't get degraded the way lactose does, so it stays in the cell longer. But part of the problem when using IPTG is it gets transported in, and it's very difficult to control. Half a millimolar to a millimolar is almost the same amount of activation of that lac operon. But by having mutants in the LAC-Z and the LAC-Y genes, altering the permeases, you get a better dose response. So a smaller amount of IPTG gives you a lower level expression. So you can tune the level of expression by dosing higher and higher concentrations of IPTG, which is really, you can do it a little bit with BL21, but not very well. And so in other words, if you want to maybe slow down the expression of your protein, because we'll learn about inclusion bodies later on, maybe you want to try the tuner so you can get slower response and then add some more later on and do all sorts of tricks with how much IPTG you get to induce this. The origami is an interesting one. This comes from a modified K12, not a BL21 strain. And these have mutations in the thyrodoxine reductase and glutathione reductase. If you hear about reductase, you should think about hydro groups and redox reactions. And really what we're talking about is the ability to properly form the cysteines are these disulfide bonds as protein folding, thus the name origami. To be able to use these cells, you have to be in AMP-resistant strains, and the Pet28 vectors are in kanamycin-resistant strains. And so if you have a problem with proteins folding because of cysteine bonds, and there's a number of different proteins that are out there that this is the case, the origami works really well for that. There is an origami V, which is both a tuner combination with the origami. And so you can then play games with how much protein you express and make sure that you get proper disulfide bonds. Rosetta is interesting. Rosetta takes the tuner-derived cells, but they add additional tRNAs. As I mentioned before, if your plasmid, the gene you put in your plasmid is not what we call codon optimized, you're going to have a problem in bacteria. So for instance, there's multiple codons for different amino acids, right? The wobble hypothesis and all that. So let's say in humans, to make glycine, maybe there's 2 or 3 different codons. And in humans, they use 1 or 2 of these a lot more than the others. So that means the codons are going to match tRNAs that are in high frequency in human cells. But when you take that gene and put it into a plasmid that goes into bacteria, maybe those bacteria don't have as many of the 2 codons that are high in human cells, but they have a lot of tRNA for a different codon for glycine. So now what happens, the polymerase goes along and is looking for a tRNA that matches one of these rare codons for bacteria. Nothing happens, the polymerase stops or terminates, and you get truncated or no protein expression. So to cover for this, there's 2 tricks around this. One, you can codon optimize your gene by changing the codon to make sure that you're expressing the correct 3 base pair sequence to get the amino acid for the tRNA that you find most frequently in a bacteria cell or host cell. And or you can use these Rosetta cells because they have additional tRNAs engineered into these strains to handle these kind of issues. Rosetta Gamma are combinations of, well, origami and Rosetta. So there's all sorts of combinations of these things. And then the Arctic Express cells. These are BL21 cells that have additional proteins being made in the cells from Antarctic bacteria. These are chaperones that allow the protein to grow, thrive, and more importantly, make protein in very cold temperatures. Because some proteins at high level of expression actually cause the proteins to be put into what we'll learn as inclusion bodies. It's toxic to the cell or the proteins misfold, and we don't want that. So maybe we want to slow down how much protein we make. Well, one way is use the tuner. So you use less IPTG and you make smaller amounts at lower rates of the protein. Another trick is to use this Arctic Express. By using this strain in BL21, you put your plasmid in this strain of bacteria and you reduce the temperature of your expression and culturing. So that way you have lower amounts of cells, slower expression of the protein, and maybe they don't go into these misfolded inclusion bodies. But to do that, you need some chaperone proteins to really make sure that things are going well. So I've talked about inclusion bodies. Let's kind of get into that. Actually, all sorts of cells, yeast, mammalian, and bacteria have what we call inclusion bodies. So inclusion bodies, kind of shown in this cartoon here, are kind of a garbage can. It's a different subcellular organelle than a lot of the others you may have learned about. And this is what happens when you get high expression rates of proteins, proteins that misfold or even toxic to a bacteria cell. They get shuttled into these very sturdy intracellular vacuoles or bodies. They're called inclusion bodies, right? And it takes a while to deposit all these misfolded proteins in there, but if that's how the bacteria is saving itself from too much protein in its soluble portion of the cell. All right, so if you get too much protein and some of your protein goes in there, in fact, even in a good expression system, we talked about watermelon and human MDH1 are good expressors, you look for the inclusion bodies, you'll still see a whole bunch of proteins there, and they do not lyse with cells. They're very difficult to lyse. You have to use strong detergents and chaotropic salts and things like this to break open and dissolve those inclusion bodies. So they're pretty much no good to us. Plus the fact that the proteins are aggregated and misfolded in these inclusion bodies, they're not going to be functional. They're not going to have the right structure. So for some folks that just can't get around the inclusion bodies, and that's why we had all those different cell strains we just talked about, for some folks in the biotechnology industry, biochemistry use, they'll actually go ahead and let the proteins express at high levels, isolate the inclusion bodies, dissolve or break apart the inclusion bodies, denature the proteins, and then refold them either by doing it in stages fairly quickly, allowing the protein to fold into its correct final state, or to use a dilution or addition of a chaotropic salt like urea to finally get the protein to fold. And each one of those is a trial and error kind of way to do it. And it's very effective if you're good at it. It takes a lot of time to do this. To be honest with you, most of us try to avoid getting to this step and use all those other tricks of different strains and different helpers and times and temperatures and ways of culturing the cells during inclusion to avoid having to do this. But you should know about inclusion bodies, and that's why we have all these different strains to try to avoid their production. So now we know we've got the right strain, whether it's for DH5 alpha or something for DNA work, or it's a BL21 or one of these other odd strains for making protein. Now we got to get the DNA into the bacteria cell. That process is called transformation. It's a fairly simple way to introduce a bacteria cell or DNA into a bacteria cell. It's really 3 steps. We take the bacteria and mix it with the plasmid DNA. This bacteria, to be able to take in the plasmid DNA, they have to be made competent. And there's a couple different ways to do this. There are electrical ways to break open or put fissures in the cell wall, cell membrane, so that DNA can then, through diffusion, get inside the cell, let the cell membrane then seal itself and capture the bacteria, or the plasmids that have gone in the bacteria. Chemically, it's mostly done with the divalent cation like rubidium chloride or calcium chloride. That causes or pokes holes in the cell membrane and wall that allow for a short period of time the cells to be porous to the plasmid DNA, where the plasmid DNA will then diffuse, driven through osmosis. As the cells warm up, the cell walls and membranes start to reseal up, capturing the plasmids inside. So that uptake takes some temperature and what we call heat shock. All right. The efficiencies of these competent cells range, and we'll talk about that in a little bit. But when we talk about the efficiency, it talks about how much of the plasmid gets into the DNA. All right, so you got to select the right bacteria, BL21, DH5 type. You have to have ice buckets, tubes, and incubators set up ahead of time at 37. You have to have a water bath at 42 degrees for the heat shock. You'll need your plasmid DNA, competent cells, and as you'll see when we talk about the protocol, they have to stay on ice and cold until you're ready to use them. They start warming up at all, they start sealing, they're no longer competent, and you're wasting your time. Depending upon where you buy or make your competent cells, you'll need some sort of a media, SOC, SOB, very similar to each other, NYZM or LB media. All of these can work at different levels. Typically, we use SOB or SOC media, LB plates, and the appropriate antibiotic. And then finally, we have an outgrowth state. And the idea is that your plasmid has not just the gene you're looking at, but a gene that will make a protein to degrade an antibiotic. That way, any bacteria that doesn't have the plasmid won't be able to grow, divide, and create daughter cells in the presence of that antibiotic. Only those cells that have your plasmid are going to be making a protein that degrades that antibiotic, thus they can grow. So antibiotic resistance isn't magical. It means that there's an enzyme being produced from some gene that the bacteria got. That enzyme will grab onto one of these antibiotics and degrade the antibiotic. So kanamycin and ampicillin are 2 common antibiotics that are used. They work through very different pathways. Kanamycin block the RNA processes. And so that requires an outgrowth. So what is an outgrowth? Outgrowth means that after you got the plasmid DNA inside the bacteria, you got to let the bacteria grow for a while without antibiotic. And so this outgrowth allows the expression of whatever genes, especially the resistant proteins that we're talking about, these antibiotic degrading enzymes that are encoded in the plasmid backbone, so that that enzyme is there and can start breaking down, hydrolyzing the kanamycin, so the kanamycin doesn't stop the messenger RNA growth. And because you're affecting messenger RNA growth, cells just won't grow and divide when kanamycin is around. It needs some time to make that enzyme before they grow. But ampicillin inhibits enzymes of the bacteria that allow for cross-linking the wall. So the cells can grow and divide, they just have very weak cell walls, and they don't need so much time to make that. They can grow and divide as the enzyme that's coated in the plasmid that will bind to and break down the ampicillin is made. So if you're doing ampicillin resistant clones, you don't have to do the outgrowth. If you do, it doesn't hurt. But kanamycin, you have to have the outgrowth because you're affecting the RNA processes versus the cross-linking the membrane, where cells can still grow and divide, where they won't if you've mucked around with the RNA processes by kanamycin. So some key important things to think about transformation is that how efficient are those competent cells you're using? And you can buy these or make these, and they're very expensive to buy and they're fairly cheap to make. But the more expensive the cells are usually mean because you have high efficiency. The high efficiency just talks about how many bacteria or how many plasmids will get inside the bacteria and how many of those bacteria will be able to take up and then continue on propagating with the plasmid inside. So low competent or low efficiency competent cells are what we call subcloning efficiency or levels. And that's good enough where you have plenty of DNA, they're higher concentration, you're going to get, even at low efficiency, you're going to get plenty of transformed cells. But if you're doing something like an site-directed mutagenesis, where you've done PCR or you've done ligated work, where you only have a small amount, very dilute amount of plasmid DNA that you're trying to get inside the bacteria, transform in the bacteria, you want really high efficient or ultra competent cells. And so the two don't, you can use high for everything, but they're really expensive and they're a little bit tricky to keep competent. Another thing to think about is if you're doing PCR products or ligated samples, you need higher competent cells versus if you just have lots of plasmid DNA, you use the subcloning level. I always like to have the students, after they do their outgrowth and dilute with LB or SOC, to take that volume, it's around 1 ml or so, and to take a small amount and put it on one plate and spread that, and then spread the rest of it, a large volume, in another plate. Because day to day depends on how well you did your transformation, how much DNA you had, the quality of your DNA, the quality, the efficiency of the competent cells. What you're trying to get is an isolated batch of colonies on this and not a lawn. So if you did a good job, that small volume that you add of the transformed cells, you'll have individual colonies. And in that case, the large volume, it'll be a whole smear or a lawn of cells, and they won't be isolated from each other. In other words, each individual cell needs to lay down and create all of its daughter cells to get a mound of cells. And you want those to be individual because that's a single colony. But what happens if the cells weren't really efficient because they were old, or your DNA wasn't at the concentration you thought, or it wasn't a high quality DNA? Well, then by doing the high and the low volume thing, the low volume, you might not get any colonies, and the high volume, you might get a few. So it always makes sense in our rule of thumb is to do a high and a low. One's going to be right, the other one's going to be unneeded, and you can just toss. It's interesting, Agene talks about, and Agene's a great source for a lot of information and little helpful hints and Power Pro tools or pro techniques and tips. They talk about if sometimes actually using less DNA, not so much the volume, but the less amount of DNA can somehow increase the number of colonies if you have problems with some of your transformations. So on our website, I'll show you that in a second, we have the general workflow or the protocol for doing transformations. And it's really, the most important point is written here. Do not let the cells warm up. You take an ice bucket with you to the -80, you take your cells out and put them immediately in the ice. Don't walk around with them, don't set them down, don't let them even begin to thaw out. You take your cells, put it in there, put it in pre-labeled and pre-chilled tubes. You don't want to hold them in your hand, and don't let them fully thaw out even at room temperature. All right. And so the protocol you can see is written here. What I'm going to do now is I'm going to stop sharing for a second and share screen Safari. And so here's the website that has protocols and stuff. And there's a background video for those who don't remember the plasmid video. It's what is a plasmid here. And from Plasmid 101, from Plasmid Adgene is a handout right here for you to review. The transformation protocol that I just showed you is linked here. And then there are two different videos. One video made by an undergraduate student in my lab a few years ago. And then the video I'm going to show you right here from Adgene.
Transformation is the process by which foreign DNA is introduced into bacterial cells. Plasmid transformation is an important technique for microbiology experiments because it allows plasmids to be stored and replicated in bacteria. Scientists use genetic modifications and chemical treatments to create bacterial cells that are easy to transform. These are known as competent cells. In this video, we will show you how to use a standard transformation protocol to introduce plasmid DNA into competent cells in order to store and replicate your plasmid of interest. At the end of this video, we will talk about two alternative protocols that can significantly reduce transformation time or improve efficiency. To start the transformation process, thaw an aliquot of frozen chemically competent cells on ice for 30 minutes. There are several different types of chemically competent cells available commercially. You can also make your own using commercial kits or do-it-yourself protocols available online. Once acquired, competent cells are usually stored at -80 degrees Celsius. Multiple freeze-thaw cycles can reduce competent cell viability. To limit competent cell exposure to multiple freeze-thaw cycles, you should aliquot your cells into small volumes prior to storage and only thaw the amount required for your experiment directly prior to performing your transformation. As the competent cells thaw, place the appropriate LB agar plates on your bench. These plates should contain the antibiotic that your plasmid of interest will provide resistance to. The plates will reach room temperature by the time we're ready to spread our transformed cells. Next, add the plasmid DNA to the competent cells. Add between 10 picograms to 100 nanograms of plasmid DNA to your cells. Mix the cells and DNA by gently flicking the tube with your finger. We use DH5 alpha cells for most of our transformations because they are efficiently transformed by many plasmids. As a control, you should also mix a separate aliquot of competent cells with water and carry these through the rest of the transformation process. These cells should not grow once plated, but will give you confidence that your antibiotic works properly. Be sure to carefully label your samples and controls so you don't confuse them along the way. Next, heat shock the DNA competent cell mixture by placing the tube in a floating rack inside a 42 degrees Celsius water bath for 45 seconds. Then place the tube on ice for 2 minutes. This change in temperature increases the permeability of the bacterial cell membrane and allows the plasmid to enter the cells. After heat shock, we allow the transformed cells to grow for a short period of time in a shaking incubator. During this outgrowth step, the transformed bacteria are incubated in a rich medium like SOC, which contains glucose. This brief period gives the bacteria time to generate the antibiotic resistance proteins encoded in the plasmid backbone. This step is not as critical for plasmids containing ampicillin resistance and can be skipped in instances where ampicillin is used. To start the outgrowth step, first add 500 microliters of SOC to the tube containing the transformed bacterial cells. Next, place the tube in a shaking incubator at 37 degrees Celsius for 45 minutes. To increase the number of colonies you get after plating, you can concentrate your cells after the outgrowth step by centrifuging. We usually centrifuge the cells for 5 minutes at 4,000 RPM or 1,500 Gs. After you've removed some of the supernatant, you can resuspend the cells and pipette them on top of a warmed LB agar.
Just as a note that the centrifugation is especially helpful if you have low confidence cells or very small amounts of DNA, so your efficiency is really low. For your average plasmid transformation, that isn't necessary and that can be skipped, and that's not involved in the protocol that you can find on the website.
Containing the necessary antibiotic. If you use pipette tips that are easy to bend, like ours, you can also use them as spreaders when plating the cells.
Notice they did the bending on the upturned inside edge of the plastic, not on the bench, not with their fingers, and more importantly, not on the agarose itself. This is a great way to make a plastic hockey stick.
When bending the tip, and make sure you are using good sterile technique throughout the process. The last step is to place the agar plate into an incubator for overnight growth. While most strains require incubation at 37 degrees Celsius, some may require lower incubation temperatures, such as 30 degrees Celsius.
Notice they incubated these plates with agar up and facing upside down, if you will. That is so that way any condensation that happens doesn't get onto the agar plate if it was upright. So incubate your plates upside down and well labeled.
After overnight incubation, inspect your plates for visible colony growth. Remember, your control plate should have no visible colonies present. If your sample plate has colonies that are too close together, you can pick a clump of cells and streak for single colonies on a new plate. See our Streaking for Single Colonies video for more information. Although this protocol will work for most plasmids, it can be shortened for easy to transform plasmids or altered for plasmids that are difficult to transform. If you want to significantly reduce the time needed for transformation, skip the heat shock and outgrowth steps entirely and simply incubate the DNA competent cell mixture on ice for 5 minutes before plating. This could impact your transformation efficiency, but will work for samples that are easy to transform. You might not always get transformants the first time you try. This could be due to the reagents you use or the size of the construct you are transforming. You should check that your competent cells are still viable and that you use the right antibiotic. If your construct is larger than 10 kilobases, it is a good idea to use electric competent cells and an electroporator instead of heat shock. Unlike chemical transformation, which relies on heat shock to cause the cells to take up DNA, electroporation uses an electric field to induce membrane permeability and often leads to higher transformation efficiency.
All right. So you saw in the video the basic steps for transformation. Both videos that I showed you on the website have this basic protocol in there. So this will be the end of the first of 3 videos to talk about bacteria protein expression and purification. Thank you.